Hey, my name is Abigail. So today we're going to be going over the enzyme aldolase, its different isoforms, and how each of those isoforms are going to vary in their expression. So to start off with, what is an aldolase? An aldolase is a glycolytic enzyme that's classified as a lyase. It's capable of catalyzing reversible cleavage and formation of carbon-carbon bonds. And so it plays an essential role in efficient energy production and carbohydrate metabolism. And it does so by having a crucial role in breaking down sugars, um, which those products can then go into energy production. So it can be found throughout the body, but predominantly in the muscle, the liver, and brain tissues. And it's found virtually in all organisms. So it can be subdivided into two classes. Class 1 aldolases are going to be found in vertebrates, plants, and green algae. And then class 2 is going to be found in bacteria, fungi, and some archaea. So those are going to differ mainly in their reaction catalyzation mechanisms. Class 1, what it does is form covalent shift bases and does not require metal cofactors. Class 2, bacteria, fungi, and archaea, requires divalent metal ion cofactors to stabilize its intermediates because it doesn't form those shift bases that class 1 does. So one of the bigger differences is going to be the state of its intermediates. And then it can be found in multiple different isoforms, the aldolase enzyme, and each of those is going to have distinct tissue distributions. And then even though all of them are going to catalyze the same biochemical reaction, the way that the different tissues are distributed uh, is specific to meeting the specific metabolic commands of that demand, sorry, of that tissue. And then, so understanding these different isoforms is going to lead to some insight on the importance of like tissue specialization, differentiation, and why it's important to metabolically regulate for efficient energy production and use of food sources and sugars, things like that. So cells require a constant supply of energy to perform their essential functions. Glucose is one of the primary energy sources for most organisms. Glycolysis is a process that's going to convert the glucose into pyruvate, which is necessary for producing ATP. And then in the gluconeogenesis portion of it, it's generating glucose from non carbohydrates such as lactate or amino acids during periods of fasting when there's no carbohydrate sources coming in. And so aldolase plays a pretty important position in both of these reactions because it catalyzes a specific reaction that gets used in both of these pathways. So what aldolase does in glycolysis, at the position of the fourth step during that process, it catalyzes the reversible cleavage of fructose 1,6 biphosphate into two different 3-carbon molecules. So you can see that in this chart here. It's on the left side of that arrow. There's two different 3-carbon molecules. In the first stage of glycolysis, the cell is phosphorylating and priming that glucose to be divided. Then the aldolase comes into a pretty critical point to cleave that carbon-carbon bond and to split that fructose ring that you can see on the right side of that enzyme into two distinct pathways, and that's going to be DHAP and G3P. So if it splits into the DHAP, it cannot continue down the glycolytic pathway because DHAP is going to be a ketone sugar, and a ketone sugar is not able to oxidize the way that it's necessary to move on in glycolysis. So in the next step of glycolysis, it requires an oxidation of an aldehyde, so it can't use the ketone sugar that way. So a DHAP has to be converted into a G3P before it can proceed to the next step of glycolysis. If it goes down the G3P route, it can just continue. And so once we move into gluconeogenesis, the part where glucose is being synthesized from non-carbohydrates, it's catalyzing the aldol condensation of DHAP and G3P. So through this process, it's creating a fructose 1,6 biphosphate product. And this is where we can see that the process is reversible, so it's able to cleave and form those carbon-carbon bonds. This is where we're able to see that, where we see a new product of the fructose 1,6 biphosphate. So the three isoforms of aldolase that can be seen in vertebrates are on three different aldolase genes. That's going to be aldo A, aldo B, and aldo C. So A is going to be located on chromosome 16, B is on chromosome 9, and C is on chromosome 17. So they're independent, unlinked locations on different homologous chromosome pairs. And that's what's consistent with early vertebrate genome duplications, also known as tetraploidization. So it's thought that these different isoforms arose through duplication and divergence. Gene duplication is what allowed aldolase proteins to evolve specialized functions while maintaining their original catalytic activity. So they're pretty highly conserved, especially in their active site and the way that they fold molecularly, as well as just with their primary like catalyzation mechanisms. So it's seen that 65 to 80% is shared amino acid sequence identity between each of the three isoforms. So genetically, they are identical, they're the same, even though they're on different chromosomes. And they do, for the most part, perform the same function, but they've specialized into different tissues. So despite their location and their functional divergences, and this is something that can be referred to as subfunctionalization, and that's something that'll come up a little bit later on in the presentation as well. This is basically an evolutionary process where a duplicated gene that before on its own performed multiple different functions mutates so that the two resulting copies are going to divide that original workload. So that work is still getting done by now, but now it's being kind of like subdivided between multiple different like entities. So despite its location and those functional differences, it's pretty highly conserved for its original catalytic mechanism purposes. A little bit about the structure of these different isoforms. So A, B, and C are going to share highly conserved tetrameric structures and that original catalytic mechanism. Each subunit of the enzyme is going to contain a conserved catalytic site. That active site contains one lysine residue that forms that Schiff base intermediate that we mentioned earlier. And so while the overall structures are very similar, 3D wise, small sequence differences that can be seen in the amino acid, because if you remember, we said it was about 65 to 80% identical in their amino acids, so there are some subtle differences. And so these are going to affect enzyme stability, substrate interactions, and regulatory behavior. But these differences are also going to contribute to their specialized physiological roles that they've specialized to contain over their periods of duplication and divergence. So the first isoform is going to be aldolase A, which is encoded by the gene aldo-A. This is going to be the most ancestral form of aldolase compared to the other two isoforms, and it's also the most widely expressed isoform. So it can be mostly found in skeletal muscles, sometimes why it's referred to as the muscle aldolase, and also in red blood cells. Because skeletal muscle is frequently experiencing like rapid fluctuations of energy demand based on your activity level, aldolase A is going to be abundant in these tissues. So, and it's going to be tissues that, as we mentioned, see high glycolytic activity. Because aldolase A is able to support rapid ATP production in those tissues. But despite that, it does have other functions that it can be responsible for. And this is a process that can be known as moonlighting. So it's moonlighting for other functions. This can include cytoskeletal interactions, cell growth, proliferation, maintaining the cell shape. Sometimes even it's been seen migrating into the nucleus to aid in DNA repair. So for aldolase A's expression, we're going to see that it is definitely the most widely expressed isoform out of the three. It's primarily seen in developing embryos and adult skeletal muscle. So in skeletal muscle, it can be up to 5% of the total cellular protein to support the striated muscle contractions and ATP production. And it's going to be the primary fetal isoform across most of the developing tissues. It can be seen in other highly metabolic tissues, such as the cardiac muscle and the erythrocytes, which are red blood cells, but it can also be seen in areas like the brain and nervous system, and it might even be seen at levels that are roughly equal with aldolase C, which is the brain-specific isoform, which we'll see a little bit later. And it can be found in some other tissues also at lower levels, but for the most part, it's going to be predominantly in developing embryos and skeletal muscles. And something that's interesting about aldolase A, it actually, during fetal development, aldolase A is going to cover most developing tissues. Um, and then once tissues move into like processing sugars, processing nutrients, aldolase A is going to get repressed in the liver and kidney in adults, and that's where aldolase B is going to take over, which we'll talk about in a little bit. But that kind of shows you also that idea of subfunctionalization that we talked about. So these isoforms are going to be performing the same functions, but they're going to be taking over certain tasks from one another. Altogether, they're doing a full body system function, but they're just going to be doing slight bits of it on their own. So aldolase A is frequently overexpressed in various tumors for various different kinds of cancers. Tumors that have high glycolytic activity, and in these disease states, cancer cells are going to be relying on aerobic glycolysis, which is something known as the Warburg effect. So the broad expression of this aldolase A reflects a full body need for glycolysis, and its especially high abundance in the muscle and red blood cells correlates pretty well with the energy requirements of those tissues, which are pretty high. The next isoform, aldolase B, which is encoded by ALDO-B, and this one, unlike aldolase A and C, has evolved to play a major role in fructose metabolism. And this specialization is pretty important, specifically in the liver, where dietary sugars are processed and distributed throughout the body. So it's expressed in organs that are involved in nutrient processing. So that's going to be the liver, the kidneys, and intestines. So the primary role of aldolase B is to split a fructose 1-phosphate. So you can see in that diagram on the right here, it's going to show you those different pathways of aldolase A and then aldolase B and C. So what aldolase B does is catalyzes the breakdown of fructose 1-phosphate into usable energy substrates, and that's going to be DHAP, and in this case, it's going to be glyceraldehyde instead of G3P. And in this case, that product, DHAP, can either enter directly into the glycolytic pathway to produce energy, or it can move into the gluconeogenic pathway to produce glucose. And then that glyceraldehyde product can be converted into G3P to be used in energy pathways. So, due to this, that puts aldolase B pretty much directly at the crossroads of glycolysis and gluconeogenesis, which in this case is going to make it important for glucose and fructose homeostasis. As we mentioned, aldolase B is primarily going to be expressed in the adult liver, kidney, and small intestine. So it's correlated with tissues that are involved in nutrient absorption and metabolism. So in the liver, the highest expression is going to occur in hepatocytes. It's a central player in breaking down that fructose 1-phosphate into usable energy intermediates. That's going to be the glyceraldehyde and the DHAP, like we discussed previously. And then in the kidney, it's going to be expressed in the renal cortex to support renal gluconeogenesis and fructose metabolism. And then in the small intestine, it's going to be expressed in intestinal mucosa to handle dietary fructose absorption and the initial processing of sugars. So because of this, because of its locations and the tissues it's involved with, it's going to be heavily regulated by different dietary carbohydrates, how much it's expressed. And then, as we mentioned a little bit briefly while we were talking about isoform A, so during embryonic stages, isoform A is the primary, the predominant isoform. And then after birth, the expression of aldolase A is repressed in these organs, the liver, kidney, and intestine, and aldolase B is going to be induced. But there are certain cases in certain pathological states that B can be repressed and A can take over again. And we touched on that isoform A can be overexpressed in cancer cells sometimes. So that's the case if in liver cancer cells, B can be repressed and A can take over again. The last isoform is going to be aldolase C, which is encoded by ALDO-C. And this one can be referred to as the brain aldolase. It's primarily localized in the central nervous system, so it can be found in neural tissues. And particularly, this is going to be within the astrocytes, the hippocampus, and the Purkinje cells of the cerebellum. So, what an astrocyte is, it's a star-shaped glial cell in the central nervous system that acts as essentially a support system for the brain. So, it's going to regulate the environment around a neuron, maintain the blood-brain barrier, supply energy to the neural cells, and it's going to manage neurotransmitters. So, unlike aldolase A, which primarily functions in glycolysis, um, and gluconeogenesis, and then B, which is specialized to fructose metabolism. C is going to drive canonical glycolysis and fructose metabolism, and it's going to do it all for the neural tissues. So, since the brain consumes a large portion of the body's glucose, despite only being a small fraction of the total body mass, aldolase C is specialized to these neural tissues to help meet these energy demands, and it contributes to the specialized metabolic processes that are taking place in neural tissues. So something interesting about C is that it actually has a similar efficiency to aldolase A, um, but where we saw that aldolase A is very widely expressed, anywhere from skeletal muscle cells to cancer cells, C is going to be specialized to these neural cells. And that's because C can focus on driving that glycolysis and fructose metabolism to support the entire neural system instead of kind of dividing its workload up again. So aldolase C is going to be highly specialized. So it's going to be expressed solely in the nervous system. And it's going to be primarily localized in that way to the nervous system and neural tissues. In the cerebellum, it's expressed in parasagittal stripes, those alternating bands of positive and negative expression within those Purkinje cells. In the hippocampus, expression is heavily concentrated in specific CA3 neurons. And then it can be found in moderate levels detected in the cerebral cortex, the spinal cord, the olfactory bulb, and then it's widely expressed in those astrocytes that we mentioned earlier throughout the entire central nervous system. And it does see a limited expression in the retina and the inner ear. So it's going to see expression in all major retinal cell types. And then in the inner ear, it's going to be found in the sensory epithelium of the inner ear and the ampullas of the semicircular canals within the inner ear. And so, because it's highly concentrated in very specific regions, it can be used as a biomarker for certain brain cells. Researchers and pathologists use aldolase C as an indicator to map different cerebral compartmentalization and to identify astrocytes. And a thing to note, special thing to note, is that aldolase C is not going to be uniform throughout the brain. So certain neuronal populations and regions are going to exhibit particularly high levels of aldolase C. And so what this suggests is that there are specialized metabolic requirements throughout the nervous system. So in those places where you're seeing high metabolic demand, you're going to see a higher population of these aldolase Cs. So that's why it's important to be able to use aldolase C as a biological marker. So you can identify compartments of the brain, compartments of the nervous system that are producing a lot of energy and consuming a lot of energy. Now that we've gone over those 3 isoforms, just to summarize everything up a little bit, this comparison in this chart here is going to highlight basically the major theme of the presentation, is that all 3 of these isoforms are performing the same enzymatic reaction, but each of them is expressed in distinct tissue distributions where their specialized properties are going to be able to provide the greatest physiological benefit for that specific tissue. And so that all brings us back to that idea that we've discussed a couple times, subfunctionalization. These 3 isoforms are going to be filling in gaps for one another, and they're going to divide up this total workload to aid in full body energy production and metabolism. So you can see that here where we've got the different levels of expression of each of these isoforms, basically by organ, by tissue type. So if you notice, there's not a single organ tissue type where you've got high expression of two different isoforms at the same time. So in skeletal muscle and in the heart and in the erythrocytes, you've got high aldolase A. In the liver and in the kidney, you've got high aldolase B. And then in the brain and the retina, you've got high aldolase C. So they're dividing up that workload pretty much evenly. And that's going to allow each of these individuals to kind of maximize the work that they're able to do. So this is going to detail on the topic of expression, developmental regulation. This is going to detail that concept we've gone over a couple times of where the isoform expression is going to shift during embryonic development into adulthood. So embryos are primarily expressing aldolase A. It's expressed pretty much throughout those tissues and serves as the primary glycolytic enzyme throughout fetal development. So then as tissues are maturing, a programmed shift is going to occur. In the liver, aldolase A that we've mentioned before is progressively repressed and replaced by B, which becomes the dominant isoform right after birth so that it can begin to handle those dietary fructose from incoming food sources. So as we move into adulthood, we're able to see where everything kind of moves from A into different differentiated tissue-specific isoforms. So on the topic of their molecular regulation, each aldolase gene has its own promoter. So for isoform A, it actually functions under three separate promoters. So pH, PN, and PM are going to be its individualized promoters. And so as we've detailed kind of how aldolase A is a lot more widely expressed, it moonlights, has a lot of other functions. It basically can be thought of as it just has a little bit of a bigger workload than the others. So it kind of makes sense that it's got more specialized promoters that are controlling its expression and its regulation. B and C are both going to be operating under one promoter each, but that's going to be controlled by specialized transcription factors. For example, some of the ones that are listed here. So expression is going to be responsive to various environmental factors like nutrient availability, hormone signals, oxygen levels, and cellular stress. So one of those to note here is oxygen levels. So each of the isoforms can be directly induced under low oxygen conditions, commonly through hypoxia-inducible factor 1 or HIF1. That's basically going to allow these isoforms in these genes to be upregulated in scenarios where there's low oxygen. So in tissues that are oxygen stressed, these isoforms can still be expressed to help with breaking down sugars and creating energy. So then although each of these isoforms are performing similar enzymatic functions, their expression is controlled independently by these different promoters. This is going to allow different tissues to adjust their aldolase levels regardless of the others according to the changing metabolic needs of that specific tissue. So what's the clinical significance of these isoforms? So as we mentioned a little bit briefly, they can be used as biomarkers for certain conditions, or even just to track cell types, basically. So through a blood test, it can detect, measure, and monitor certain conditions. This might be things like muscle or liver damage or general inflammation. So under aldolase A, the expression of that isoform is going to be elevated in times of muscle injury or damage. So using it as a biomarker will be able to lead you to those areas of injury. It's also increased in several cancers, since as we talked about, there's an overexpression of aldolase A in certain tumors. And aldolase B, so B is tightly linked to numerous different inherited metabolic disorders. One of those is hereditary fructose intolerance, which is caused by mutations in the ALDO-B gene. And these mutations basically destabilize the tetrameric structure of the enzyme, rendering it basically completely inactive and causing severe toxicity upon fructose ingestion. So that fructose is going to accumulate and basically be toxic. And then in aldolase C, it can be used as a biomarker for neurological injury. It can be sometimes as a diagnostic marker in plasma or cerebrospinal fluid for conditions that range from congenital disorders to certain types of lung cancers um, and neurodegenerative diseases. And then, as we mentioned, it can also be used as an indicator to map different compartments and identify just certain cells like astrocytes in the nervous system. All right, just to summarize a little bit really quickly. So, isoforms A, B, and C are going to share catalytic activity, but they're going to differ in their expression patterns. And that's basically kind of been like the main overarching theme of this whole thing is that subfunctionalization. Aldolase A is going to predominate in the muscle and glycolytic tissues, high activity glycolytic tissues. And then aldolase B is going to specialize in fructose metabolism in the liver, kidney, and intestine. And aldolase C is going to prioritize kind of all aspects of energy production in the nervous system. And so the three aldolase isoforms are a pretty good example of how tissue specific expression and specialization can create functional specialization in an organism. So in this case, it's going to be so that they divide up the load through subfunctionalization to basically enhance and ensure like maximum metabolic efficiency throughout the entire body. And here are my references for the presentation. And these are going to be my image references. So thank you so much for listening. All right, bye-bye.